at a different scale than we usually go.
Yes, that's true. It's in a cellular scale. I hope it's
Oh,
still
I
interesting.
just saw for the people who are local,
At a different scale than we usually go.
the picnic tomorrow got cancelled because of heat. Oh, Evie, that's a new look.
What
Never,
look?
never
Yeah,
seen the Shatterstone.
yeah, the sun. I'm afraid of the sun.
I love that you're going all
It comes
black the
today.
second the sun comes out. Right. Last Neuroccino before the summer break that apparently we can all use given that we're hiding from the sun and canceling meetings because it's too hot. Um, today, I know, you're just laughing from Spain. It's hot by Dutch standards. Pedro, take it away.
Okay, so
And
good
if
morning.
you could
I'm
share
going
the link in the chat, please. That'd be great.
the link of the article.
Yes.
Okay, uh, just a moment.
That's a well-hidden article.
Okay. So may I start?
Lovely. Thank you. Now, if you share your screen,
Yes.
ideally in full
Oh,
screen, and then we can take it away. Perfect.
okay. So good morning. So the article that I'm going to present today was published in Science in March and is entitled Synaptic Structure of a Memory Engram in the Mouse Hippocampus. So as Stephanie was referring, this is a different scale that we usually work, but still I hope it's interesting. So basically the topic of this article is memory engram. This concept refers to the network of neurons that are activated when we encode and when we retrieve a certain memory. And this network formation, the main mechanism or the main model behind this mechanism is the Hebbian theory. So this well-known theory states that when two neurons have similar functions, when they fire together, they also wire together. That means if one neuron provokes the activation of the other neuron, this co-activation tends to strengthen the synapse. So these synapses tend to be stronger than the synapses between two neurons that do not co-activate. And this article explores the cellular mechanism behind this formation of networks at the cellular level and presents some conflicting or at least complementary evidence to this Hebbian theory. So basically, the authors used a mouse model and they studied these neurons in the hippocampus. So we have CA1 and CA3, and they studied the synapse between between a CA3 neuron and the CA1 neuron. So they are studying the connection between the axon of the CA3 neuron and the dendrite of the CA1 neuron. Basically, this area that I'm showing here in the picture. What they did was that they use a virus, and this virus induces a genetic modification, and this genetic modification is related to this FOS gene. This FOS gene basically is a marker of neuronal activation. So whenever a neuron is active, the neuron quite quickly starts to increase the expression of this gene. So basically it works as a marker of neuronal activation. And with this genetic modification, this FOS gene leads to is the production of another protein, and this protein is very unstable, so it is degraded very quickly. The interesting thing here with this mechanism is that we can inhibit whenever we want the degradation of this protein. So when we administer this drug, the trimethoprim, we can inhibit the degradation of this protein. So if you have a neuronal activation, this leads to increased FOS gene, increased level of this protein, and without trimethoprim, the protein is degraded. When the authors gave trimethoprim at the same time, the protein is not degraded anymore, and it leads to the production of another protein. And this protein has fluorescence properties. So this protein can like be a marker in this case, electron microscopy. So the experiment was based in a behavioral paradigm. So basically, as I told you, the mice were injected with this virus, and then they were trained in a memory condition. In this case, in a Pavlovian paradigm. So basically, they associated certain visual or olfactory cues with shocks in mice foot. So there is a Pavlovian mechanism associating this stimuli with an unpleasant, with another, in this case, unpleasant stimuli. So in order to like enhance the creation of this memory, this association memory. So basically, when the mice were subjected to this memory paradigm, they were injected at the same time with the trimethoprim. So they could have a marker of the neurons that were activated, in this case in the hippocampus, during the encoding and retrieval of this memory. And here is an example of what happens with this mechanism of trimethoprim injection. So this is this negative paradigm without the injection of trimethoprim, and we don't see like any fluorescence signals. And this is what happens with the injection of trimethoprim at the same time of this associative memory. And you can see very well a lot of neurons in the hippocampus that are marked with this protein. And here are the first results. So as expected, so here we see this fluorescence sign, and this is the behavioral paradigm without trimethoprim. This is the behavioral paradigm, and this is the injection of trimethoprim. So as expected, when you have the injection of trimethoprim and the behavioral paradigm at the same time, you have a higher percentage of neurons in this area of hippocampus that are marked with this fluorescent sign. Basically, it shows that the experiment is working, or at least measuring what the authors tend to measure. Here is the image of an axon that is marked. So in this case, is an axon of a neuron that was activated during this behavioral paradigm, among other ones that were not activated. And then the authors used convolutional neural network algorithm to automatically characterize or to automatically identify the cellular structures in these microscopic views, because of course there are a lot of cellular structures here, and it would be much faster, and if this identification would be done automatically, and this is what they did, and of course this was validated with direct observations. So here you can see dendrites. So these thick structures are dendrites, and these thin structures are axons. This was an interesting thing that I didn't know. Actually, is this very well described before, but I have never realized that dendrites are much thicker than axons. I think actually this is usually not illustrated in pictures usually, but dendrites are much thicker than axons. And here in green you see activated dendrites, and in gray you see dendrites that are not activated, and then this in blue you have these blue thin structures are axons that are not activated, and these green or lighter blue are axons that are activated. And here is a full picture of a synapse. So this is the kind of pictures that they got, and so the blue areas are axons. These tiny yellow dots are vesicles, so the vesicles that are released by the presynaptic terminal. In gray you have dendrites, and in this I forgot the violet color you have like the dendrites buttons, okay, these post-synaptic densities that are basically the dendrite buttons. So thinking about this Hebbian mechanism, basically you can have four kinds of configurations in these connections. So you can have inactive dendrite that is connected with an inactive you can have an inactive axon that is connected with an inactive dendrite. You can have an inactive axon that is connected with an active dendrite. You can have an active axon connected with an inactive dendrite, and you can have an active axon with an active dendrite. And according to this Hebbian model, the most probable states would be these two. So then axons that are not activated would tend to be connected with axons with neurons that are not activated as well, while axons that would be activated during this memory engram formation would be connected with dendrites that are also activated. And here is the first conflicting result of this article. So according to the Hebbian theory, as I told you, we would expect that and this graph shows this. So basically what you see in green are structures that are activated. Here you have the state of dendrites, inactive or active, and here in green you have axons active and axons inactive. So according to the Hebbian theory, we would expect that this column, so when you have axon active, you would expect that the number of dendrites would be higher than the percentage of dendrites that are active with inactive axons. Okay, basically this thing of two states that I told you here, we would expect that this state would be more frequent, but this doesn't happen. So the axons that were active were not more frequently connected with active dendrites than those that were not active. So as this result is conflicting with this Hebbian theory, the authors looked into other kind of properties of these synapses. And here they described two kind of synapses. So the most frequent type of synapses are called single button synapse. So in a single button synapse, an axon is connected with one dendrite. So here you see the axon, here you see the synaptic button, and one synaptic button is associated with one dendrite. Basically, is a one-to-one relationship. However, there are other kinds of synapses. These are called multi-button synapses. And here there are two examples of these multi-button synapses. So here in this case, you see an axon, and instead of this axon is associated with just only one button, here this axon is connected with two dendrites by two different buttons, and here in this case a single axon is connected with four dendrites. Okay, so here you have four synaptic buttons. So basically one axon is connected with more than one, more than one neuron. And what the authors found, and here is a representation of, again, of these multi-button synapses, what the authors found was that the axons that were activated during this memory paradigm, they had more multi-button synapses than the axons that were not activating during this memory paradigm. So the axons activated during the memory paradigm that compose these memory engrams, they tend to connect with more more dendrites. It's a more widespread network. And then the authors also looked into the volume of these synapses, because it's described then when a synapse is more active, so that if there is more transmission between these two synapses, the volumes of the terminal axon and the volumes of dendrites increase. So, and this was what was observed. So basically when they compared like the synapses that had activated dendrite or an activated axon, an activated axon or an activated axon and dendrite, which are the green lines here, these two green lines. So both activated or activated axon and inactivated dendrite, the volumes of these synaptic structures were higher, and confirming these previous observations that more active synapses have larger volumes. They also looked into like the composition of certain organelles in the cells. So again, it is described that synapses that are more active, they have more mitochondria in axon terminal, because they are of course consuming more energy, so they increase the number of mitochondria in the synaptic in the axon terminal. And here in red you see this. So here again, this thick structure is a dendrite, this thinner structure is the axon, and these red areas represent the areas in which mitochondria were identified. And what they found was that, again, areas with activated axons have higher volumes of mitochondria than axons that were not activated. Finally, they looked also into non-neuronal structures, in this case astrocytes. So these glial cells also are involved in the synaptic structure, and they help in the different functions like promoting like the functioning of the synapse, regulating the levels of neurotransmitters. And so it's expected that again, synapses that are more active tend to be more astrocyte participation. And here again, this is the picture representing this. So here in blue you have the axon, and in yellow again you have the synaptic vesicles. In violet you have the dendrite, and here these larger structures represent the astrocyte. And what they found was that these astrocyte terminals were larger again in axons that were active. Okay, so to conclude or to present the main conclusions of the authors, basically the first conclusion is that their analysis didn't confirm this model of this Hebbian model that neurons that that fire together are wired together, because they didn't find that more active axons are more frequently connected with more active dendrites. Instead, they found that neurons that are, or axons that are activated with a memory formation, they tend to be connected with more dendrites. So they tend to create, not to like reinforce a network, but to create larger networks or more widespread networks. And at the cellular level, these synapses had like different biological markers showing that they are actually more functional active. So they have larger volumes, they had more mitochondria, and they had more participation of astrocytes. So as limitations, the authors refer that this is a snapshot. So of course, when they sacrifice the mice, they cannot see what happened during the week in which they learn this memory paradigm. So this is a snapshot. They cannot evaluate the dynamics of the synaptic formation. So they assume that actually during the first stages of memory engram formation, there can be like this Hebbian mechanism, and then this Hebbian mechanism somehow changes to this different biological mechanism of more widespread networks. So we cannot say that this is against the Hebbian theory. It can be against or can be a complementary mechanism to what is more widely accepted. And then the other big limitation is this FOS gene approach. So basically the authors use FOS gene activation as a marker of neuronal activation, and actually this is the most common approach in these studies of chemo targeting. But of course we don't know that if FOS gene marks every kind of neuronal activation. There might be some kinds of neuronal activation that are not characterized by this FOS gene activation. And basically that's it. So this was a cellular level study, but I found very interesting the way that they can characterize so well these connections at cellular level.
Thank you. Very impressive work. Hands are flying up, and I think Lilith was first.
Yeah. Hi, Pedro. Thank you for presenting this paper. I don't. Do you really think that this? Because as far as I understood, okay, so two neurons that are fired together, right? They are active. They use the same function, but they wire together. So, but in this case, it's kind of also true. Just if there are other conditions are met, for example, as you said, more connections with dendrites or so it's not that Hebbian theory is wrong, it's just more detailed conditions were described in this paper, or do I not understanding it well?
Yes, I agree. So, because the Hebbian theory states that if they fire together, they wire together. But here we are looking, or the authors are looking to mice that were sacrificed. So we are not seeing actually the activation at that time. We are seeing a marker of neuronal activation that happened at the same time that they were being submitted to this memory paradigm. So they and the authors are very clear in this, so they cannot say that the event theory is wrong. Because this learning condition happened during 1 week, and it was just after this week that the animals were sacrificed. So at the first stages of learning, there might be a Hebbian-like mechanism that we cannot assess, or the authors cannot assess when they looked at the cellular level after 1 week of learning. But if the Hebbian mechanism was the sole explanation for this uh, creation or reinforcing of memory networks, we would expect in the first result
Yeah, yeah.
that that this state would be much more probable, because here you have an axon and a presynaptic neuron and a postsynaptic neuron, and we would expect that this state would be much more probable than all the other states, or at least in these two states that we have activation of one and inactivation of the other, and this was not true. So somehow the Hebbian theory seems to not explain everything. So they state that maybe what another mechanism of like reinforcing learning is like creating more like widespread connections, not reinforcing a network in the sense of strengthening one connection, but reinforcing a network by creating more connections.
Mm, okay. But like also the concerning these results, how is it possible that an activated neuron was not connected to other activated neuron? Did I understand correctly? That
Yeah, it's somehow so. I think here we are not. So here we are not seeing in a time scale that is
Mm,
coincident with the time scale of a synapse. So here, of course, if we are seeing, if you could assess this in the time scale of a synapse activation or inactivation, I completely agree with you. So we would expect always this or this. But
yeah.
here we are not assessing at the time scale of a synapse. So I think the memory paradigm, I'm not sure how long it took, but several minutes. So basically we are seeing like the probabilities of being activated in a very longer time scale than the actual activation or inactivation of a synapse.
Mm,
That's why we are measuring like, or they are, well, of course, they were measuring like the the activation of the neuron during the memory paradigm, not during a single activation of a synapse.
okay. Thank you so much. Very interesting.
Thank you.
Eduardo also has a question for you.
Yeah, thanks, Peter, for presenting. I think it was a similar question to the one Lily asked. I'll try to rephrase it in a slightly different way and ask you whether it's still possible to save the Hebbian kind of approach. Not because I particularly like it, but because it's the established theory. So, do you think that, for example, because you mentioned that even though there isn't any functional coupling where the neuron that fires does not correlate with another neuron firing, this kind of memory formation is more correlated with the formation of actual connections. Do you think it's possible that because of the higher number of connections that a presynaptic neuron establishing with the postsynaptic neuron, then there is still some sort of space to save the Hebbian mechanism because simply having more connections, like physical structural connections to the postsynaptic neuron, is actually allowing for maintaining this kind of firing pattern. So it's like more likely that a firing in the presynaptic neuron than is triggering some sort of reaction in the postsynaptic neuron.
Yes, so so the authors they don't aim to say that now the Hebbian model is wrong. They don't mean that, and they are very clear in the discussion. But actually, they in the introduction they present other arguments that were already against the Ibn theory as the sole mechanism behind these memory engrams. They say, for instance, if the Ibn theory was the sole explanation, we would expect then when a memory is encoded, you would have like the formation of a network, and then if when during the processes of memory retrieval, you would expect the activation of exactly the same network. But previous results showed that this is not actually true. So when in animals there is a memory encoding and the network is activated when with optogenetic techniques when they study the network that was assessed with memory retrieval, the overlap, I'm not sure about the ratio, but the overlap was about 25%. And we would expect, if the Hebbian theory the sole explanation, we would expect 100% overlap. So it seems that somehow after encoding, memories, there might be some readaptation of the network. And of course, this experiment cannot show how this happens, but it shows somehow that there are other cellular markers of memory engrams behind just strengthening to synapses.
Okay, so it's
But
more
I agree
about the
that
general connectivity pattern of the entire network rather than just like a specific sub part.
yes, and one thing is that this study didn't test memory retrieval, so they just test like learning during 1 week, and then the animals were sacrificed and analyzed at that time. So they didn't test memory retrieval. They wanted to like characterize what happened at cellular level after 1 week of learning. Of course, there would be retrieval when the animals saw those cues, but
Yeah, interesting.
Does that answer your questions for now?
Yeah, a bit so. And sorry for missing the introduction.
Thank you
I
so
know
much.
actually I didn't tell that in the introduction.
Okay, then I'll say.
Just an after explanation.
Thanks so much for presenting. This is the final Neuroccino before the summer break. So enjoy your heat day tomorrow that we got in the Netherlands. And everyone else, enjoy your summer. We'll be back in September. I believe Monday is the 1st of September with Neuroccino. And then we also have the CNS talks again. So have a great summer. See you then. Bye.
Bye.
Ciao.